My name is Adam de la Zerda. We're now in Stanford University in my lab in the Department of Structural Biology in the medical school, and I'm the PI here. So I grew up in Haifa, which is a town in the north part of Israel. My father is a chemist, and my mom is an architect. When I came to Stanford, I studied quantum physics, and I never took a single biology course in my life. Literally a single biology class in my life. A good friend of mine passed away from brain cancer, and I decided to put all of my history behind, all my interest in quantum physics behind, and try to join the cancer research community. What we're hoping to get to, or our vision for the future, is to be able to create those tiny little molecular agents that are the size, that are nanometric in scale, so smaller than 1,000th of the thickness of a hair, and inject these agents by the billions into our bloodstream and have them go all over our body and go cell by cell and kind of like knock on that door and ask this cell, are you of a particular kind that I'm interested in? For example, are you a cancer cell? And if you are a cancer cell, are you responding to the treatment we're giving you, as an example? And if the answer is yes, they will bind to it. If the answer is no, they will let go. Those that have decided to bind will now start to send signals to us and say, hey, look at me, I'm here. And we'll be able to recept that with those special cameras that we're building outside of the body. And in fact, when we're looking at a tumor that is really that small, like maybe like a garbanzo bean sized tumor, in that particular tumor, you can have as many as 1 billion cancer cells. When we're actually trying to treat someone that has cancer, for us to cure that person means to kill 100% of these cells, not 99.9%, truly 100% of these cells. And that's very tough. You can see the tiny little particles we've created, and what we've done here is we've brought them to the field of brain cancer. And they were able to find the brain tumor and actually show us exactly where the tumor is. And so now, if you're a surgeon and you're trying to cut away only the tumor and leave the healthy brain intact, you can actually start doing it here. What's the most exciting part of being in this lab is that it just does, to me at least, and I think to other people too, it just doesn't feel like coming to work. It's actually an electron microscope. So this is zooming down to those tiny, tiny, tiny secret agents I was telling you about before. And interestingly enough, they've created some sort of a pattern here. This is by complete mistake, right? This is one of those failures that we talked about. And you'll notice that they actually made some interesting shape there. So
it's a heart.
Exactly. Yeah.
You think?
And that concept of work doesn't quite exist here. For us, we're like kid in a candy store. I think another thing I really like about this lab is it's very, very diverse. People are coming here from all corners of the world with so many different backgrounds. Some are chemists, some are physicists, some are engineers, some are biologists, some are medical doctors. These are really, really good people, naturally good people that actually that really care about science and actually want to make a change and affect humanity in a positive way. It's about bringing the absolute best people, but having a very strict no rule in the lab. It's just a very, very strict rule. It doesn't matter how good you are. If you're you're not. This is just, it's not going to work. Failure is probably the one single most important thing in science, and it's the thing that is so important because frankly most of the time we're failing. I would say that I tell my students that if 1 out of 10 experiments that they do actually succeeds, that's wonderful, that's phenomenal. The type of things we do here are super high risk. Of course, if we get there, there's also a huge reward, but there's so many reasons why we will fail. You have to be really open about this and say, hey, the experiment failed. It's not me that failed. I did not fail myself. The experiment failed. And now let's try to learn why is it that the experiment failed and see what can we learn from that. Most times we can learn a lot more from a failed experiment than we can learn from a successful experiment, actually. We really start from zero and we try to get here, right? And this is a huge jump already to do, and one that is not trivial at all. In fact, many times there's a good chance that there is no way to get from here to there. There's no way to get from point A to point B. In some ways it's like going to a new type of planet, and you're almost guaranteed that wherever it is you're landing, there's something interesting that no one has ever been there before, and that's the beauty of science. Now in 2015, I think medical research is probably one of the most exciting places to be in. I'm of course very biased, but I hope for good reasons. A lot of things happened over the past 50 years, and a lot of them just came to fruition recently. So the type of questions we can ask ourselves these days are questions that we never ever imagined we'll ever be able to ask. And there's this general feeling when you go to conferences now and you talk to other scientists that actually everything is kind of possible now. You have to have a very strong self-confidence in you, because most of the times you will fail. And so it's that passion for learning and passion for creating that come together with a very strong self-confidence that you know that if you set your mind to something, you will get there. If you're really passionate about something, if you really want to solve a certain problem, if you want to do something that you believe is really significant, really profoundly important to you, there's probably nothing that can really stop you from doing it.